Well, welcome to the ACID workshop for the Botanical Society of America, Botany 2021. One of the key things we're going to be talking about today is doing things simply, explaining your work in the most simple possible terms you can. Simple but technical. And I really think that Einstein is correct when he says if you can't explain it simply, you don't really understand your work well enough. I would say this is absolutely true of myself. I only really started to understand what I was doing in my work and the limitations of what I was doing and its potentials when I started giving better talks, when I started giving more simple, clear talks. So although we say it's about presentations today and it's about presenting better, it's really about understanding your work better. It's about understanding your work better and having a bigger impact on your field. And so we're going to come back to that point of having a bigger impact as we go along through our workshop. So even though it looks like we're working on titles, we're really working at a much deeper level. We're working at how you understand what you're doing and how you have impact on your field. There's a book coming out, and a lot of the things in the workshop are covered in the book. I'm very excited to have written it. I'm looking at the page proofs in early July, out in October. They say maybe late October. But the web page is up here for the book, and I will post that in the chat when I have a chance to do that, the URL for the book. So please take a look at it and consider purchasing this thing. And I think it's got a lot of good advice in it. And it is published by someone who you may not heard of before, CABI. CABI.org is an organization that primarily does development in the third world. And all of their profits go into development work in the third world. So I am just delighted that I am not publishing with Elsevier or somebody out, one of these big guys out there who is making money off the book and putting it in some fat cat's pocket. This is all going to help make the world a better place. And so all the profits go from their publishing arm go to that. It's a pretty unique kind of book for CABI. They mostly publish technical things in the areas that they work on in development, technical scientific papers and things. This is a book written by a scientist for scientists about presenting science. So please have a look at it. We hope to get some reviews out there as soon as the book is out. Okay, we're going to jump right in. I've got some titles. I've taken these titles from the botany workshop last year. I have made them anonymous. So I haven't even recorded who wrote these papers. Could be someone you never heard of, could be someone who's well known. I want you to think a little bit about which titles tell you best what this talk is going to be about. These are all presented talks, not posters. I'm going to stop talking here in a minute and give you a minute to think about that, and then I'm going to run a poll so you can do this. So you can think of respond anonymously, and then we'll talk about what your answers are and talk about these titles. So for the people who are watching this who are not present, that is watching the video, we're getting responses to this. 18 of the 21 people have responded, and that's probably all of the scientists we have. We're probably not getting responses from the Speaking Center staff. Okay, so we've got most of them for number 4, and number 5 comes in a close, not a close second, but second, and number 3 close to number 5, with number 2 not getting any of them. So let's look at number 2. I would like to know how many people know what wrangling means. I think native English speakers, someone who grew up in a different language beside English, would not know this word. And so this is a real, but for someone who is a native English speaker, and especially who grew up reading cowboy stories, it has important meanings, but it's a very obscure word. And so this is a real warning to you to be wary of those kinds of cute words in your title that are going to miss a good portion of your audience, especially if that audience is a diverse audience and includes non-native English speakers whose first language was not English. Maybe have excellent English, but you didn't grow up reading cowboy stories, of the West, and you don't know what a wrangler is. So a wrangler, there's the mythology of the cowboy in the American West is that there's this rugged individual who spends his time out riding on open plains and taking care of cattle. And that taking care of cattle is a very difficult job. They're very they're very independent beasts. They're not really smart. They wander all over the place. And that process of taking care of especially of difficult cattle, is calling wrangling them. So a cowboy who does that kind of work would be called a wrangler. And so what this person of number 2 is trying to say is that they've got this heterogeneous data and they are struggling with how to make it, make sense of it and get it to go into the corral and to get it to form a coherent story. So if you know the word, it contributes something to you, but it is a really bad title for everyone else who didn't grow up in the United States reading cowboy stories. So beware of that. Let's look at number 1. We're going to come back to number 1 later. It's got 1 vote. Niches in time, space, and environment, concepts and comparative context. So it tells you about niches. If you work on niches, you might be interested in it. And I'm not going to say anything more about this because I'm going to ask you more about this title a little bit later. So we'll come back to that one. 3, 4, and 5, you could make an argument for all of these as being the best one here. Differentially expressed genes contributing to the evolution of nectar reduced in the selfing syndrome. Okay, it tells you pretty clearly about what this talk is going to be about. Differentially expressed genes. They contribute. How do they contribute? Can you tell from the title what this research is really going to show? It's a good start. It's a really good start. But it doesn't tell the reader how the genes, these differently expressed genes, are going to contribute to the evolution of reduced nectar in the selfing syndrome. And if you could put that into the title, even if it's a bit longer, it's going to draw in more people because it's going to say, oh, I know what this is about. I want to hear this talk. I don't have to kind of worry, well, is it something? Maybe it's something. There's an alternative talk that maybe got a better title. Maybe I should go to that one because I know what it's going to be about. This one might be relevant. It might not. Let them know what it's about. I'd have the same kind of comments about number 5, revealing the dynamics of sunflower domestication with archaeological data. Again, it's pretty good, but it doesn't tell you what the dynamics are. It doesn't tell you what's going to be revealed. What is going to be said about the sunflower domestication? If you could put that in, it would be an even stronger title. So the one that got the most votes and the one that would be my choice for this is structure of inflorescence in the Lowiaceae has similarities to the Strelitziaceae and may support common ancestry. It's still got problems. And we're going to look at those next because I'm going to ask you how you would improve this title. So in order to do that, we are going to send you out into breakout rooms. Okay, I think we're back. Would someone like to share some ideas about how this could be improved?
Hi, can I?
Yes, please say your name and then.
Hi, everybody. My name is Laura Calvillo Canadel from Mexico, from the UNAM. And we discussed about the title that, for example, you could have an idea about what the talk is going to, how do you say, how the posture or the talk, el tema, but for example, the structure of inflorescence, you could eliminate maybe the structure and to write only the inflorescence in Lowiaceae, and you could supreme Zingiberalis because it's an order and it's not necessary. It has similarities to that of Strelitziaceae, for example, may support common ancestry. And the other is to write the different form.
Well, let's let someone else. You got a great one. That was a great suggestion. Let's let someone else do the next one.
Okay, thank you.
And I really like how she introduced herself, your name, your institution, and then say, go on, how would this be improved?
Thank you.
Kelsey?
Yeah, hi. I'm actually an undergraduate at Columbia. I'm in New York, and and I guess I have sort of a unique perspective because I have much more. I do science writing for the public, which means that I basically translate a lot of papers into ways that the public can understand. But I've done little actual scientific writing myself. So from that perspective, my move would be to use common names for the families. I don't know if that is good advice for writing for a scientific community, but to me as a journalist, having something to contextualize this a little bit in case you're not exactly in this field is something that I would do.
Yeah, great. Thanks, Kelsey. And I encourage you to jump in today and give a perspective on how you would make things available or more accessible to the general public. Now, this workshop is a technical workshop, and we're mostly focused on presenting to other botanists. So I'm going to stay focused like that. But it is really nice to have the point of view of how you would change something to make it more accessible to the general public, and people can also think about that a little bit if they are doing other types of presentations. So thanks very much for jumping in with that. Sylvia is next, whether she thinks she's second or not.
Okay. So I'm Sylvia. I'm a postdoc at the University of Montreal. So my idea here, I also thought about simplifying this family and order thing, because both families belong to the same order. So I think it could be like, you could remove Zingiberalis or put it more general, like two families of, I don't know. But simplify that part. But the main thing is that I think that the goal here is that the similarities may support common ancestry. So I would start with similarities. So similarities between inflorescence structures may support, and not saying that they have similarities and that these similarities support the common ancestry. You go already for the goal that is the similarity.
Great comments. Great, really great comments. Miguel, is that pronounced right?
Yeah, I'm here. So I'm Miguel. I'm a postdoc researcher in Clark University in Massachusetts. And I just realized that I have a big problem with the common ancestry thing. That is not very specific. Like probably you will want to say something like sisterhood of Strelitziaceae and Lowiaceae or something like that. I mean, many people have a problem with the Zingiberales, but I'm used to work with really weird organisms. So having like a context of like a broader phylogenetic context, I think is a good thing to have an idea of, because I have never heard about these families, but I have heard about the Zingiberales, so at least I can kind of place where the analyses are in the tree of life. So that's
great. Another more great comments. You guys are brilliant. You're doing great on this. I can't wait to see your rewritten titles. Any last comments on this before I say a few things?
I don't know how to raise my hand electronically or through Zoom.
Yeah,
so
that's working very well.
Thank you. So I'm Vera Velasco. I'm a senior research associate at University of Toronto in Mississauga. I just have one comment, and obviously it depends if the work was actually done and it's supported by data, but it would be nice to specify what the structure is, because it says the structure. So if it could be specified what the specific structure of inflorescence is, then it would be, I think, a step towards a little bit less bigness. Thank you.
Great, thanks. I really appreciate the comments and the great thoughts you guys have put into this. And now I tell you, this is one of my papers. We started out with mine just to show you that this is one of my best titles and it can still be improved. And I've made every mistake we're going to talk about today. I just wanted to demonstrate that very viscerally here. So even when this was from last year, when I started to really work on these kinds of issues and wanted to write better titles, and still I don't think I've nailed it. I got better than I have ever done in the past. It would have been some very generic, odd title you couldn't tell what was about in the past. So aside from all the comments that have been made, which are all very good, I want to talk about this word may. Here we go, working. So this is something that I struggled with a lot, and I suspect you might struggle with a lot as you start writing these titles. You're trying to put your main result into your title. And sometimes that result is not completely clear, just because data is not always completely clear. This problem of a relationship of Lowiaceae and Strelitziaceae, and the bananas, Musaceae is in there too, the banana family, and there's one other family, has been problematic for a long time, and it remains problematic. They've done whole genome sequencing and stuff, and it doesn't resolve it. It's a mess. So that was why this was an exciting thing, that there might be a morphological character that supports a relationship here in this. So may is a weasel. I call it a weasel word, right? I didn't say it's going to support the relationship. I thought about it a lot, and I said, that support common ancestry. I could have said that. I mean, you have to decide how much you are going to leave for the talk itself. Not everything has to be in the title. I could probably have said it supports common ancestry, because it does. But it is very hard to tell without constructing a whole phylogenetic tree from the ground up for every new character. It is hard to tell if that character really is a shared derived character or a shared primitive character. And I don't know for sure which one it is. And I talk about that in the talk. Now, does that mean I should say may, or does that mean I should say it does support common ancestry and leave the details to the talk? You're going to have to make that decision for yourself on every talk. And if you're like me, you're going to change the title 5 times before you submit it. But I wanted to point this out because it'll probably start bothering you as you start to write titles that more succinctly deal with your main result. Are you really representing your data or not? So just remember in that context that you don't have to have everything in the title. Your title is to get the main result there and to get people in to listen to you so you can deal with all the complexities that you want to tell them about. So again, great feedback on this title. Really appreciated it. I really like some of the comments, and if I had to do it again, I would change some of the stuff that you all recommended. Let's do it again. Here we have another set of titles. I'm going to let you read them for a minute, and then I'm going to start the poll and let you decide if there are 1 or 2 of them that you think are particularly good. So again, number 5 came out at the highest level. And then 2 and 6 had some votes, and then the rest are pretty low, with 1 and 4 getting only 1 vote each. So you did such a great job of talking about how to improve that last title. Let's go down these and tell me a little bit about these titles and why you find them informative or not informative. We'll just start with number 1. Let people chime in as they feel so inclined.
Okay, I have to discuss the geography of Penstemon.
Yes, we'll just do them in order.
Yes, you could know about, okay, that the talk is about Penstemon and the distribution of Penstemon, but we need to be more specified which part of from which country, or compared with what areas, for example, I think.
Yeah, so the title Biography of Penstemon doesn't really tell you anything about what the person's going to say. It just doesn't tell you what their conclusions are going to be, anything about this. just as Laura has just said, it tells you the general title. Are there other comments about biogeography of penstemon? One person liked it if they're willing to say something.
I have a question on this. So wouldn't exercises like these have a tendency to be biased against descriptive talks rather than those that like actually have a true conclusion, like you did an experiment and then you have results to talk about?
So descriptive talks, I work in a descriptive field. All of my work is basically descriptive. The work that I talked about in the last one was descriptive work. I put it into a phylogenetic context. So with descriptive work, you have to ask why you're doing the work. Why was this an interesting question? What is it contributing to science? If there's no contribution to science, if it's basically you're just satisfying your own interests, I think we are at the place in science where we have to ask, why are you doing that work? Certainly, you're going to have a very, very difficult time getting a grant for it. You're going to have a difficult time getting tenure, maintaining an academic position, if the only reason you're doing it is to satisfy your curiosity. If there's another reason that you're doing it, and there almost certainly is, put that in the title. So it's not, I don't think, as a person who does only descriptive work, I don't think there's a bias against descriptive work, but it forces me to say, why am I doing this work? Why is this an important question? Why was it important to describe flower development in the Marantaceae? Why was that, and what was that going to contribute to scientific knowledge? That should be in the title.
So you would have to give whoever presented this title would have to give some sort of hook to say, well, what makes the particular biogeography of penstemon stand out from any other biogeographic example for any other genus or any other family? For example.
For example, thank you for saying for example there, because we're going to come back in a minute to titles that are like this one and ask you some questions about them. There are reasons why you might write a title like this. But for what we're doing right now, what you said is right. For example, they would have to say something about, why is penstemon important? Why is this biogeography important? The biogeography of penstemon demonstrates, something. Tell the audience that why it's important, that they should care.
Cool. Thank you.
Let's go on to title number 2. People have comments on that?
Sure, I have something to say about this one, I guess. This one was my second choice because I thought it outlined the results fairly well. But then for me, the term performance seemed kind of vague. And that's why I decided it was my second choice and not my top choice.
Yeah, I'm with Emma. Oh,
go ahead.
I'm with Emma about performance. I also think that sounds vague. And I would also love to see this result seems pretty not very surprising to me. And I would love to know if there is a specific finding that is a little more surprising or interesting. I would love to see that introduced in the title as well.
Yeah, so Kelsey used a really important word in that, and I don't want that word to go by without notice, and that word is surprising. So in the best title, in the best presentations, you are going to surprise your audience. And here is the really good news. All research is surprising. You just have to emphasize it. We will come back to this in a little bit when we talk about the structure of a story and how science stories fit into classic story structure. But all research is about finding something new. And if you find something new, you have overturned previous ideas, and that is surprising. So I really want to emphasize the fact that you should be thinking about what is surprising in your research, which is the same thing as what is new about your research, which is the same thing as what is the most important point about your research. But if you can get that across to the audience, the surprise, you're doing great.
Well, I was just thinking that Douglas fir is not a real fir, so hyphenating that would probably be good. So I guess this is pretty elementary for everybody, is to make sure that you have the right spelling and the right stuff. Yeah.
Yeah, it's very important in scientific when you're talking to technical audiences or professional audiences to have the spelling right, to have the terminology right. If you're using jargon, which even for technical audiences I don't recommend, you have to have those things right. Since the word audience has come up, I don't know, I should say, I don't want to distract us. I'll save audience for later. If you want to talk about audiences and how you would adjust your titles, things for different audiences, ask me about it later. We are already dealing a little bit with some of Kelsey's comments about a general audience versus this audience for the Botanical Society, which you all know very well. We'll just leave it those two audiences for now. How about number 3? Any comments on, does earlier flowering impact plant community seed set?
Okay, it's a comment. It's more of a question because this was my third choice. So I had 5, 2 as my second choice. And then this one, I like titles with the question because I feel like inclined to watch the talk and see, oh yeah, does it? Or, but I know that this wouldn't apply for all types of work. So but I kind of like it because I'm curious now to know if it does impact or not.
So if you had 2 titles, 2 talks, competing times, in rooms next to each other, well, let's say in rooms way far away, so you have to choose one or the other, you can't jump between them. One of them has the title, does blah, 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 and the answer could be yes or no. And the other one says, this is what I found. Both are in your research area. Which would you go to?
No, I would go to the one that it's certain that what they found.
And that's what we're trying to do, because you want people to go to your talk. You don't want them to make the decision to go to another talk because it's got a clearer title. So even though, what Sylvia says is correct, these are interesting things. And if that's the only talk you had to choose from, you'd go to it. But that's not the only talk you have to go from. You've got to choose between 5 or 6 or 7 different talks at the same time. So make sure that they're going to your talk because they know what it's going to be about and they want to hear it. Number 4.
I'm Claire from University of the Bahamas. This is actually my first meeting. We have bad weather, so forgive me for not having on my camera. For me, this is kind of how I write my titles, so I quite like this title. It tells me that I'm looking at the structure, what part of the structure of the leaf, and what specifically the venation of that leaf. So that kind of homes me in to what I want to hear about. So I thought that this was a pretty good title.
I think it's a pretty good title. I wish the species or what kind of plants we're talking about is in the title. That would make me run across the conference center in order to see it or not.
Well, almost no one voted for this title, but we've only have positive comments about it. Anyone have something to say that would make it better or what might be not as good about the title?
I guess I have something to mention about this one. It kind of goes back to the descriptive title without a purpose. I think when I read this title, that maybe having some more reason for why this is important might help strengthen the title.
That would be my comment. Why is this important?
I think it's really, I'm sorry.
No, please go ahead.
I think the title is really vague, is kind of general, and it looks like the author didn't know exactly what he found or she found. It's really like in a circle, no getting to the point, and that's why I didn't like the title.
Yeah, the same. I mean, I have many titles like this. Most of my titles are like this, so I completely understand, and I see their weakness now. I just wish someone had done this workshop with me back when I was a graduate student or a postdoc. I'd be writing better titles and a lot more interest in them. Okay, number 5. Number 5 was the winner. What did people like about it, and could it be improved?
I guess this was my top choice. I thought it was concise and straight to the point, and that's why I liked it.
So the only thing that it doesn't do is tell you why it's important. And so here's another very difficult thing that you'll have to deal with in your writing titles and writing your abstracts, and that is how much background knowledge can you assume from your audience. So Iosoides is a pretty interesting genus. It's got an interesting history within the Lycophyta, and people who know it will know that this might be an interesting talk just because they know Iosoides. And if you can assume that, then the title is probably fine. If your audience doesn't know Iosoides or doesn't know whatever your group was on, right? So I worked on the Loiaceae, which is a very obscure little group of 20 species at most in Southeast Asia, very restricted distributions in the Zingiberales, but it's important for the evolutionary history of the group. And basically no one's heard of it except people who work on Zingiberales phylogeny. So you've got to explain something different if you're working on that group than if you're working on Iosoides, I think. So again, something to keep in mind when you're writing your titles. Think about your audience. You'll notice that all of the things we've been doing are directed at getting your audience more involved in your work. A lot of these titles are about what I want to say. I know my work real well, and here's what I'm saying about it. I'm going to talk about the biogeography of penstemon. I'm going to talk about stressors. They're not about what your audience needs to hear. So one way to think about what we're doing here is switching the focus from me-centered, me, what do I want to say, to audience-centered, what does the audience want to hear? What does the audience need to hear to understand my talk? So you're telling them in your title what they need to understand your talk. And you're telling them up front so they don't have to read the abstract. They don't have to do a lot of thought about it. They can say, oh my God, I know what this is about. It is of interest to me. I'm going to that talk. And if they have a choice to make between talks with have vague titles in it, right? So there's an open time in your schedule. You haven't got something in there, let's say, well, let's see what's going on at this hour. And you've got a talk that is going to tell you what it's about, and you've got 4 talks that are kind of vague and you're not sure what you're going to about, they're probably going to go to the talk where you know what it's going to be about, if it's even of vague interest to you. So audience-centered is what we're really doing. So there's a lot of value to changing your titles in the way that I'm recommending here. Now, for this last one, I made a separate slide for this, but look at this title, and can you tell me something how you would improve it? How would you make it better?
So one thing I really like is the specificity. It tells me exactly what I'm looking at this 1893 Rydberg expedition. I think the word that I'm most fixated on in this title is the value. You can have valuation in different ways, and it's like a teaser, right? Like, although I guess it works both ways. Like, you read the title and you want to know how this is being valued, why that is important, and it may motivate you to be interested in the talk or look at the poster. But also, like, you're not able to tell why this particular expedition is important from the title by itself.
Good, thank you. Other comments?
Yeah, I was going to say that the words before the colon, I think, frame the point of the talk very well. It's going to be about why we should value our smaller collections. And as someone at a university with a very small collection, I appreciate that.
So for me also, the value of small collections, that was a really something very interesting in here, because I wasn't sure that these small collections really had very much value. Unlike the first speaker, and I wasn't sure who that was, I found the 1893, or maybe it's very similar to his comment. Rybert expedition was very specific, and I found it a little too specific here. I mean, for me, anyway, the value of the title was about small collections. It didn't really matter where these type specimens come from, but that they found new type specimens in a small collection, and they, that's really important, I thought. So I'd make it a little less specific if I was doing it by taking this information about the Rydberg expedition out and emphasizing the newness of the type specimens and keeping that stuff before the colon, as Tammy said, which frames it very nicely. And let's think about that question I started to ask about earlier about these talks like biogeography of penstemon and this talk about niches in time, space, and the environment. Now, I have a poll that I've created for this, but I'm also, maybe we can just do it because there are such a small group that we can just ask you to climb in and tell me why you would go hear this talk. We know it's not specific. We know it's not, you don't know what this person's going to say in detail about niches. Why in the world would you go to this talk? It's not that you wouldn't go. There are some good reasons why you might go to it. Why would you go to it?
I think that the words concept and comparative context will give, since it's not my area, I would feel like, okay, they're going to explain to me what they're going to give me the basic concept, and then I'm going to be able to follow this, and this would be interesting.
Okay, so if you knew nothing about niches and you wanted to learn something about it, it seems like it might be a general talk that was going to teach you a little bit about niches, niche concepts. Okay, that's a good reason. Other reasons?
I like the time, space, and environment, even though that there's some vagueness and broadness in the title. I would go expecting to see and take away useful comparisons between the three contexts that are provided for exploring the concept of the niche.
Yeah, again, so a general talk about niches that's going to give you a good overview of the talk. So, yep.
I think I would like to see how all these three factors overlap and they give me a better sense of the niches.
Yep. Okay, same kind of things. We're getting the similar comments said in different ways. Let me start the poll here and give you some hints why I think you might want to do that. Let me find the right poll. Got 13 respondents, and maybe we should stop here. But anyway, so there's a lot of reasons, right? These are reasons why you might go to the talk. It was a part of a symposium, and that also means that the author was better known than other people who give a contributed talk at the workshop, and you might want to go to hear that. I mean, I know that there were people in my field, now retired, who always had big numbers of people at their talks because of who they were. It didn't matter what the talk was on. People knew it was going to be a good talk because they knew who was giving it. Didn't matter what the title was. They went to hear their talk because they knew the person who was doing it. If you knew the person giving this, you know they give good talks, you're going to go to hear it. You have to ask yourself, of course, with your talk, whether that's you. Whether you, at this stage of your career, are well known enough that your talk is going to attract people because it's you. I, at my end of my career, still wonder about that. I kind of doubt that people are coming to hear the talks because it's me. So I am starting to focus us with this question on a really important concept that I want to end with before we take a break. So what's the one thing you want your audience to remember about your talk? And it's the same answer for everyone in this room or in this workshop. But there is one thing, and it is really important. And you'll see the segue from our last question. That is what I believe you want them to remember. I believe it is the only reason you go to conferences. It is the only thing you're trying to convey. It is the most important thing. Because if there's a job opening and they're saying, well, who works in this field? They want them to, you want them to remember Sylvia. You want them to remember Jose. You want them to remember Miguel. You want them to remember Mariana. You want them to remember you. You want them to remember you and your work. And so there's a number of ways that you can get that. One of the ways is the way we've been talking about it. And the real reason we've been talking about this rewriting titles is so that they will come to your talk and remember you. Because when they have a job opening, when they have a postdoc open, when, you want them to think about you, when they have a collaboration open, when they need someone with your expertise, you want them to think about you. You want to get to the place where you can write a title like the niche concept in ecology. That's your title. And people will come to hear it because it's you giving the talk. Not because it's a good title, it's a terrible title. But if they know you and they know that you've worked on this for 20 years and you have something to say about it, they'll come to hear it because it's you. Now, a question has maybe occurred to you. We are talking all of this stuff about your title, and your title is written. There are plenty of people who have a different title on their first slide and in their abstract. So if you can't go back and do it in your abstract, you'll do it next year. You'll write a better title next year. But if you can't do it, then you come and you go and you put it on your first slide. And somebody will remind, maybe will notice that difference, but it'll still improve your talk because you'll be telling them at the beginning of the talk what your talk is about. So I really encourage you to do that, to go back and change your title. So we're going to take a break, and I'm going to ask you during the break, we'll take 10 minutes, and I'm going to ask you not just to take a break, but to think a little bit about your title and think about how you would rewrite it now that you know these basic principles of put your main result in your title. And then you can come back, and if you're comfortable, you can share some of those titles with the group. You would share the before and the after title. And we'll see where we are and see if there's feedback from people on your title. We're small enough to do that. And so we have some people who maybe have rewritten their title or want to talk about the process, and let's begin with that. Was it Sylvia who had something?
Yes. So I'm not rewriting yet, but it's just I wanted to share that when I was preparing my talk, then preparing the slides, I realized that I couldn't fit in the slides everything I wanted to share about my work. And when I prepared the talk, I realized that the title should be different. And that's what I'm thinking about now. So I think it's also something that helps because you won't be able to talk about everything you want, everything you did. And this also helps because we have the deadline for abstract submissions. Sometimes we don't think about it and then we have an idea in mind. But when we start digesting the work a little bit, it changes. Yeah, but I still don't have because my work, it involves a method. So it was my approach that enable me to extract some kind of data, but the results are more or less descriptive as well. So that's where I have a hard time finding the main message, because I'm basically describing that, okay, the new live imaging method allows me to generate this type of data, and that's it. So for now,
there you go. There's your title.
Yeah.
I mean, I don't know the details, but so a new live imaging method produces something data more useful for this purpose.
Yes, what I posted on our group, it's basically, so my title is the original one. Live imaging uncovers cellular growth patterns shaping Arabidopsis stamen. And my main message was live imaging provides a quantitative atlas of cellular growth dynamics in Arabidopsis stamen. So basically, the live imaging method allowed me to generate this atlas. And
okay, but you said a minute ago that your work was about a method. Yes? Does the method apply to Arabidopsis only?
Well, what I could do, yes, but we could extend this, extrapolate this to other species with some limitations, but yes, we could.
So there's two different things here, and then you're right, you're struggling with it. I see that, and that's good. But there's two different ways to frame this, but you really framed it, I think, correctly already. it's about a method that you happened to apply to Arabidopsis. It is not, from what you said, about Arabidopsis, and here's a new method to make Arabidopsis clearer. You see that how those are two different things.
Yeah.
So you have to decide which is this. Is this an Arabidopsis talk that's only going to really be interested to Arabidopsis people? Or is this a methods talk that has broad applicability and you just happen to do it on Arabidopsis?
Yeah, that's it.
Okay, then if that's it, why even mention Arabidopsis in the title? It's just happens to be that you use Arabidopsis. Everyone uses Arabidopsis. You want people to come in to learn the new method. So tell them this new method is going to do something for you in your research. Even if you're working on the Zingiberales, it's going to do something for you. Whereas if you say it's going to do something for you if you're working on Arabidopsis, you're going to get a small audience. That's only going to be interested in Arabidopsis.
I think it's not so broad now because it's limited to model species still. But I think that the information we get from this could be extrapolated from, but I'll have to think.
Yeah, think about that. I mean, there's 100 model species now, where it's not Arabidopsis is no longer the only one. So it's still thinking about framing that. So I'm encouraging you all by talking to Sylvia about this title. I'm turning you all, go for the bigger audience. If your research has applicability to people outside your narrow field, and you are giving it at a general symposium, go for that bigger audience. Make it relevant to that audience. If you're going to an Arabidopsis symposium, then focus it on Arabidopsis and what this technique is going to tell them about Arabidopsis. But, there's going to be a relatively small percentage of people at the botany meetings doing Arabidopsis work. And you don't want just them on your talk. You want to have other people as your talk. So
I would say my focus here is the stamens, and independently if it's Arabidopsis or no. Yeah,
even if it's, it may not, I don't know the technique, but maybe it's not applicable just to stamens. Maybe when there are small carpels, there's plenty of species with carpels that are absolutely tiny that have a single layer of cells in their gynecium, and if it's got those kind of limitations, maybe it could be used there too. Or maybe there's even model species like that. There's some aquatic model species. Maybe it could be used there. Don't sell yourself short. Let your audience decide if this is applicable to them. You tell them what you can do with it. This is all about model stuff and about techniques papers now, because we're going to talk about other kinds of papers in a minute. But about techniques papers, cast it broadly. Let your audience decide if it's relevant to their stuff. Don't you decide that for them. You talk about the limitations of the technique. If it's about a technique, you talk about the limitations, and they can decide whether those limitations make it not useful for their work or not. New techniques are really exciting, and techniques that let you do new kinds of things, show them that kind of stuff. It's great. People will be very interested in that. Whereas the people who are interested in a new kind of Arabidopsis atlas or something, it's going to be very limited by comparison.
Thank you.
Other folks want to talk about their title?
Hi. I find writing titles very difficult. So my original title is Impact of Combined Heat and Drought on Water Content, Photosynthesis, and Chlorophyll Fluorescence in Interior and Coastal Douglas Fir Provenances. So obviously that misses all the important points that the title should have. Most. So I don't know, but right now what I'm thinking of is maybe something like Douglas fir can acclimate to long-term drought or extreme heat, but not under conditions where drought and heat are co-occurring.
Yeah, and that's what you said when you wrote your most important point you wanted people to remember. And so you've taken that most important point and moved it to the title. And I think that is exactly in the right direction. I think that's exactly how you should be thinking about it.
Thank you.
Yeah, you can still play with it a little bit, to fine tune it and things, but that's the right direction. Now, the other question I want to ask you is, and I think I know the answer to this, but I'm still going to ask it anyway because there are other people listening. Is your result specific to Douglas fir, or is it something that generalizes to other species? So let me give some background. So there's an example in the book about, in one chapter, we talk about titles quite a bit. And there's a title about alpine ibex and interbreeding within the alpine ibex. And it's pretty clear that the authors think that the study that they were able to do on this population of alpine ibex, where there's a huge, there's so few of these animals left that they were able to track them over hundreds of years. They know the breeding history for 100 years or something of these guys. And so the work that they were able to do on that is not just applicable to the alpine ibex. It's applicable to special kinds of situations, but it tells us things about population dynamics that we didn't know before. And so the question in those cases becomes, do you put alpine ibex in the title, or do you cast a wider net? And that's something that only the author can decide on that, which they do, but it's something that you should be thinking about. Now, I think that in Vera's case, it's specific to Douglas fir, is that right?
Yes, I think so. But your point is really good. I think it is because of many reasons. We're looking at different provenances and how differently they respond, right? And if you compare, like, I can try to go broader in terms of like conifer instead of just Douglas fir, right? But then Douglas fir is kind of an outlier in the conifer world, if I may say.
Yeah, we'll continue to think about that question, because if you can cast it broader, you'll get a bigger audience. And then in the talk, you can say, what you've said to me, Douglas fir, we did the work on Douglas fir. It's an outlier. We think that these results would be applicable to these other species, but maybe not these other species. You can get into the nuances of the differences, but you've got the audience in there listening to you then, and they won't care that, that your title said was more general than that, because you've made it clear in the talk and in the abstract what the nuances are.
Okay, thank you.
Okay, good afternoon again, everyone. So I'm working on a forest recovery project, and my current title is Citizen Science Forest Recovery and Community Perspectives on post-Hurricane Dorian Grand Bahama. So in listening and taking in all of this information, I feel that I'm missing the mark in terms of the community's perspectives on the forest recovery efforts. And even though the citizen science initiative is important, I don't want it to be lost that it's not just general perspectives on overall recovery, it's the recovery of the efforts being put into forest recovery versus social recovery. So I'm still trying to tweak it. I'm not really sure.
I would say, again, focus on your major result, your scientific result that's going to be interested to people. How you got that result in this audience, kind of a technical audience that's relatively close to your area, but isn't exactly in your area. I mean, you may get plant morphologists who come to your talk. They're not going to be community ecologists. And so in this audience, how you got the result is probably not important for the title, that it was used with citizen science. Now, when you get into the talk and you're saying, I got these results and these results are cool and this is why they're important, and we did this all with citizen science, well, that's just all the more interesting then to the people who are there. But get them into the room first by telling them what the stuff that, the real results, the takeaway message that you want to convey about what you found, and then tell them this really cool way you do it, and they'll think you're great, because not only did you have a good result, a surprising result, something new to tell them about, but you did it with a really unique way that most people can't wrangle that method. I said we look at some things from nature and science. I've really just got examples from nature. I've got 2 slides. We're not going to spend much time on this. But these are all pretty good titles. Statin drugs might boost healthy gut microbes. You know, the might there is a bit of a problem, but you can understand why they said that. It was like the word may in my title. But still, they're telling you exactly what it's about. Patterns and trends of North Hemisphere snow mass from 18. Okay, they didn't tell you here. This one isn't quite as good. It doesn't tell you what the trend is. Right? If they said patterns and trends, or decreasing patterns and trends of northern hemisphere snow mass from 1980 to 2018, that would tell you a lot more about what they're going to say in this talk. Right now, you just know they're going to talk about these trends. So even the addition of one word here would help. Ligand-induced, yeah, I can't pronounce this, monoubiquitinone, ubiquitination of BLK1 regulates plant immunity. Clearly not in my area, but it does tell you what the talk is about or what the paper is going to be about. It doesn't tell you if it upregulates it or downregulates it, but that probably is not important here. It may do both under different kinds of conditions. It tells you clearly, if you were in this field, what this paper is going to be about and what to expect by reading it. Structure of, again, I can't pronounce all of these words. They are way outside my field, but again, it doesn't tell you what the structure is, but it's telling you that there's this new structure that they're going to be talking about in the paper. It's not bad. This one's much better. Statin therapy is associated with lower prevalence of gut microbiota dysbiosis. So on these kind of technical audiences, they're writing in the best journal in the world, and they're only going to try to attack the people who are interested in that topic. They're not interested in attracting a general audience. They're interested in attracting people who know about statins and working on statins. So these very technical titles are fine for this journal. It's all about the audience. Last one, construction of a human cell landscape at single cell level. Again, it's really kind of a methods paper, but they've put it in a way about human cell landscapes on things. So they've got this, it's not exactly a new method. They've gone through and they've created an atlas out it. So I don't know what to say about it. It does kind of tell you what it's about. You know, you like them. You can't write a title like this, but you'd kind of like them to say, we constructed a new atlas of human cells at the single cell level, and this is useful in. If you could do a title like that, it would be even better here because it would tell the reader what to expect when they read this. Where here you've got to go and you've got to read the abstract to find it. And who the hell has time to read abstracts now, if it's out, especially if it's a little bit outside your field, right? So another reason to do these really clear titles is because everyone is just as busy as you are or more busy. And if your title isn't clear, they're not going to read it. They're not going to take the time to read the abstract. Get them in by having a clear title and telling them they should be interested in what you're doing. Okay, we're done with titles. So what's the story? What's the structure of a story? And why should you be telling stories in your presentations? So there's kind of a negative connotation around this word story in science, because stories can be fanciful constructions. But we are not using it that way. We are talking about true stories. We're talking about writing narratives that are true to what you have found, that are true to your research, but use a storyteller's sensibility in a way. And let's talk about what that means. Before I go on, though, I want to acknowledge my contributor on the book, John Wagner, who drew the illustrations. You'll see 2 of them in the next couple slides. This is one of them. I'm just really excited to be working with John. He's a fantastic illustrator. It's not his primary job. He works in water conservation out in Oregon. I met him at a conference and I was just delighted with his drawings. I love them. So how do you tell stories that are true? So what's the story of a structure? What's the structure of a story? So stories have been told forever, since time immemorial. And we find in the story, we have some kind of a hero. And the hero leaves their community. So there is some kind of existing community. And there's some kind of a problem. So B. And that problem causes the hero to leave the community, to go out on an adventure. And in that adventure, they have to meet adversity. There be dragons. They have to fight those dragons and conquer them, and they come back with rewards for their community. So this is the hero's journey. And we see it in all kinds of films and all kinds of stories. So in Star Wars, we have the character, the hero, Luke Skywalker. He's got some problems. He's impatient. He's impetuous. He wants to escape his uncle's farm. His uncle's farm is part of that pre-existing world. He's on Tatooine. He wants to get out of there. He wants to be a space pilot. He's very unsettled. And so he leaves. He's forced off. His uncle dies. He's forced off Tatooine. He meets a trainer, Obi-Wan Kenobi, and that changes him. He does some kind of training, changes him. And then he's got real problems. He has to face the Death Star. It's activated. Obi-Wan gets killed. The Death Star is turned on the Rebel base. All kinds of other stuff happens. After the Death Star is turned on the Rebel base, Luke must finally master his impatience. He has to learn to trust the Force, and he has to destroy the Death Star. And so that's the reward for the community. So he comes back with a box of gold in the terms of he's destroyed the Death Star. So here's the basic structure, and next slide we'll relate this to scientific stories. So here's what we've just said, a flawed character and their world. So the world is there, and there is some flaw in that world. In this case, I said the character, but there's a flaw there. That flaw is going to drive the character out. It's kind of what the whole story is about that flaw. The character is confronted with a new situation that changes their view of the world. The world now looks different because of this something new comes about. And that challenges them to take action. So the center part of the story is they're doing all this action. The Death Star is destroyed. Not the Death Star. The Death Star destroys Leia Organa's home planet, yada, yada, follows him to the Rebel base, etc. That's all in the middle. And in the final part, the character confronts the new situation and is forced to change to save the day. So how does this relate to science? It does. The Act 1, the first part of it is the current state of knowledge. What exists in your field before you did your work? That's that community of people sitting there not pushing or saying goodbye to the hero. In many ways, you can look at yourself as that hero then. That hero, you, is confronted with a new situation. Something happens. There is some conflicting evidence out there that says this view of the world is not complete. Science is always about what's wrong with our current theory. How do we test it again, or how do we find some little hole in it that we can say it's incomplete? That conflicting evidence leads to your new hypothesis. Doesn't have to be formed, hypothesis does not have to be formed strictly in the sense of hypothesis, but it is the spur then. That conflicting evidence spurs you on to do some work because you want to solve that problem. And you've got some new idea. Think of your new hypothesis as your new idea. And the main part of that then is the data collection. And that gives you the data collection. That's really the main part of Act 2. That data collection then leads to your results and the conclusions. And we already talked about science is always about surprise, because that new result overturns the current state of affairs. It changes the world. Every discovery changes the world. Every part of your research changes the world. You don't think about this, you don't give yourself enough credit. I don't give myself enough credit, but everything I've done, everything that you have done has changed the world, even just a little bit. And because it's changed the world like that, it surprises people, because it says the current state of knowledge, the current way we think the world is not the whole story. Here's something new about it. So there's a really great connection between the classic story structure and how science is done. And we need to start telling making our scientific presentations follow this structure, because it is going to be much easier for people to understand, and it's going to be much easier to give the talks, and they're going to be more interesting. So here it is, just in other terms, what is already known, the current state of knowledge. That's Act 1. That's the first part of your talk. Here's what has been done about the phylogeny that includes the Lowiaceae and the Strelitziaceae. Here's X's work, here's Y's work, here's all of this stuff, and no one has been able to solve the problem. The conflicting evidence in that case, in my case, is there's no clear answer to what the phylogeny is, and they've thrown tons of data at it. So let's look and see if there's something in morphology. Let's do the new hypothesis let's see if there's something in morphology that's going to tell us about this. And inflorescence structure has always been suggested in this group as being important. Is it? Let's see. Spend a bunch of time. You wouldn't believe how much time it takes to do this work in morphology. All science is like that. That's the data collection. I did all of that work. I don't have to repeat it. I don't have to lead them through, troubling through the undergrowth in China, almost spraining my ankle to get the species of Lowiaceae so that I could look at the inflorescence structure, sitting in a camp up in an area of China where they're doing they don't need to know that stuff. They don't need to know any of those things about it, but they need to know that there was data collection there and what I found, and that leads me to my results, the resolution of the story, the results and conclusions at the end, which then change the current state of knowledge. Okay, we want to do an example. I'm going to do an example from the book, and I know you can't read this. I have broken every rule of a good slide by this one. I'm going to read it to you, and then I'm going to show you how we're going to present this in a 3-act structure. And then I'm going to ask you to do the same thing with another example. And we can do it in your groups, or maybe we'll just do it individually here. So you get a little bit of practice of thinking about how you would break up some information, some already pre-digested information, into a 3-act structure. So I'm going to read this to you so you don't have to read it yourself. Dr. Peter Vischer and his colleagues were interested in the oldest life on Earth, microbial communities that existed for over 1 billion years before there was oxygen in the atmosphere. These microbes secreted calcium carbonate and created layered limestone rocks called stromatolites. Because they existed billions of years ago, no one knew exactly how they captured energy from the sun. There was no oxygen, so they couldn't have used oxygenic photosynthesis or aerobic respiration to store or release energy. When Dr. Vischer's team found arsenic in fossil stromatolites, they speculated that the ancient microbes may have used different kinds of arsenic as an electron donor in photosynthesis and an electron acceptor in respiration. They wondered if similar modern-day communities also used arsenic without the presence of oxygen. If they did, the team had evidence for their theory. To test this idea, they looked for living stromatolites in the extreme conditions of the high Andes. In a small stream deep in the Atacama Desert, they found bright purple microbe mats that thrive in the complete actions of oxygen and build stromatolites. Just as the clues they had found in ancient fossils suggested, these microbes used 2 different forms of arsenic to perform photosynthesis and respiration. Their discovery offers the strongest evidence yet for how the oldest life on Earth survived in a world without oxygen. So we certainly can find, already we can find some parts here, right? So there's the conclusion, there's Act 3. Their discovery did this. And when we've got Act 1 up here, there's mostly Act 1, roughly there. And then the middle would be Act 3, Act 2. So let's put it into that structure. I've broken this up just slightly differently here than I just outlined, but there's Act 1. The bacterial mats exist in the ancient Earth and they built stromatolites. We've known that. That's the existing knowledge. We've known they're there. And then some conflicting evidence is found. It's not really conflicting in this case, but you'd say it's new evidence. I struggled a long time what to call this because sometimes it's just new and sometimes it's actually in conflict. I called it conflicting evidence. You know what I mean here. Arsenic's found in the ancient stromatolites. They're not doing this with oxygen. Maybe they're using arsenic. So there's a new hypothesis. Arsenic is maybe but at least a electron donor and acceptor in those ancient stromatolites. And they do this data collection then. They go out and they look at these bacterial mats in the high Andes and they investigate them. They actually do a lot of experiments here. This is just a very abbreviated part of their research. Their conclusion then, it changes the way we see these stromatolites. We now think that they are cycling arsenic. They may cycle other compounds too. There are other compounds that existed at that time that they may also be using as electron donors and acceptors, but it sure looks like they were doing arsenic as one of them. And that's something new. So, you know, it's very clear. It follows the structure. It gives you this kind of 3-act structure, gives you a way of organizing your talk. And there's very little about methods in there, which we'll come back to. So I do want to emphasize that a bit. There's very little about methods, which means that you're concentrating on what the important stuff is to your audience. Unless your audience is working in your area, is doing exactly the same kind of work that you are, they don't care about the details of your methods. Now, there are people that care. There are people that care a heck of a lot. If you're a student, your committee cares. Your committee really cares. And they want to know all of those details. And if you cannot omit detailed descriptions of your methods when you're talking to your committee or to your advisor, it's got to be there. People within your narrow specialty. Let's start as narrow as possible. People, if you're in a big lab, people working in that same lab, they want to know your methods. It's just one step down from your committee. They want to know that. They want to know that they're being done right. Just slightly wider, people in, let's say, in an Arabidopsis meeting, in a Drosophila meeting, right? And they want to know your methods. That's a very narrow audience in your discipline. People at a general botany meeting, those people, most of the people that talk are not going to be in your narrow field, in your narrow discipline. Unless you're giving a methods talk, like Sylvia is. This does not apply to her talk. Unless you're giving a methods talk, those people don't care about your methods. They care about your results. They care about your methods a little bit. So what we are talking about here also when we talk about this botany audience is the imposter syndrome, right? You feel like, I feel like, everyone feels like. You got to go out there and prove yourself, show that you know these methods so well that you're qualified to stand up there and talk about it. And that is the wrong audience to do that to. The botany audience is the wrong audience to prove that to. You do not have to prove yourself to that audience. The fact that you got into this meeting, that's proof enough. 99.9%. In fact, in all the times I have gone to meetings at botany, and I've been going since 1981, basically to every meeting except one, I have never heard a talk from someone who I thought was unqualified to be there. I had one talk in all of that time. And it was just a meeting it was a talk that did not belong in the botany meeting. I don't know why he was there. So you deserve to be there. You do not need to prove that you're there. You need to prove to your committee if you're a graduate student. Damn right, you got to prove to your committee if you're a graduate student. You do not need to prove to this audience. You do not need to prove that your methods are right. You do not want to be in a conversation with this audience about your methods unless you're giving a methods paper, because they might find a hole in it. And if they find a hole in it, they're going to say, well, I got a job opening, but I think I'm going to overpass Emma because Emma made that mistake in the method. She doesn't really understand her work very well. So before you go in, you make sure that it's right. You convince yourself, you get people in your narrow audiences to listen to your stuff, to know that your methods are right, to know that your conclusions are right. And then you focus on those conclusions when you're in the talk. And you will get a lot of attention because you're going to be telling people what they want to know. They want to know what you found. They want to be surprised. They want to know what's new. That's what we go to the meetings for, what's new and to meet people. If the methods are new, then you got to tell them the methods. You're doing a methods paper, that's a different animal. Okay, I've been very strong about this because I listen to a lot of talks, especially by students and young postdocs that concentrate on their methods. And I think it's the wrong thing to do. I don't think it's going to get them, it's not going to give you the intention that you want. Okay, I want you to do this. Try one yourself. This is a harder one. I will read this to you as I did last time, and I will let you think about it after that. I'll stop the video, let you think about it for 5 or 10 minutes, we'll see. And then I'll ask you if you can find the different parts of it here. It's already a little bit pre-digested. Find the 3 acts here and see how you would organize this into the 3-act structure. So I'll read it here so you don't have to read it yourself. Dr. Rita Vetter and her colleagues used plant and animal toxins to understand the molecular pharmacology of pain. Given this interest, it is natural for her to work on the stinging trees of the Australian genus Dendrocony in the Urticaceae, which produce painful and persistent stings. These compounds are responsible for these stings. The compounds responsible for things reside in the epidermal trichomes that cover the surface of the plant. The pain usually lasts for hours, but painful flares can continue for days and sometimes weeks. Based on what you knew about the pharmacological active compound in the trichomes of the related stinging nettle, Urticaceae, it's reasonable to assume that this compound, a small molecular neuropeptide called rewritten, would be found in the trichomes of these species. However, human tests with the purified extracts of rewritten had previously shown that it was unlikely to be the main culprit for long-lasting pain. When rewritten was injected alone, it failed to produce the substantial long-lasting pain that occurs with contact with the plant. Also, when our team tried to isolate rewritten from Dendrocony excelsa, they found it wasn't present. These results suggested that there was unidentified pharmacological active compounds in Dendrocony trichomes. To test this hypothesis, they isolated and purified the molecular constituents of Dendrocony excelsa trichomes and assessed the sensory effects of the resulting fractions. Mass spectroscopy of late eluting fractions showed the presence of a mini protein stabilized by disulfide bonds. Experiments with synthetic versions of these peptides showed that they can reproduce the symptoms of a sting. The severity of these reactions suggested that the peptides might be neurotoxins. Hand-cleft electrophysiology of mouse dorsal root ganglion neurons confirmed that the peptides work directly on voltage-gated sodium channels, confirming this hypothesis. Though this class of neurotoxins were previously unknown from plants, they are similar in structure to neurotoxins found in spider and cone venoms. Understanding the mechanisms by which these molecules affect ion channels and nerve cells may provide new ways of controlling pain. So take a minute or so, minute or two, and think about where the problems are. Okay, someone want to tell me what the background is? What's the pre-existing world? What's the knowledge that we started with? Act 1.
I can go. I said, first off, I was very general and said toxins can be used to understand pain. And then dendrocyanide produces painful and persistent pain, likely caused by pharmacologically active compounds that are in the trichomes.
Yeah, so you've done a little more than just introduce describe the world as it exists before the research. You've also suggested a way to open it up. That is, and I think that's important when you're thinking about how to present your talk. You want to frame your talk around the most important point you're going to make about this. And for Dr. Vetter, the most important point is how we use this information about these stinging trichomes to understand human pain. And that's how Emma started out that first sentence. So if you were giving a talk, you would elaborate on that a little bit. If you were doing another thing we talk about in the book is we talk about how you'd introduce yourself in a networking situation like that. And Irina Vetter, I think, would introduce herself by saying, her name and that she works on plants that produce toxins in order to understand human pain responses. And that would be her tiny little introduction to herself. So that's a really good thing to think about when you're beginning a talk. What's the big picture that your work is turning in? So let's go on and think a little more then about that beginning. What's the world look like before she does her work? What do we know before she does her work? Emma's already given us some of it.
I would say that this genus, those plants, they cause the sting. The compounds are found in the epidermal trichomes, and the pain could last for some weeks. Could be long lasting. I would say
it's horrible pain in this case. Yep, those are all things we knew before she did the research and what attracted to her this plant. Now, there's one other thing we could say we knew beforehand, or was a reasonable assumption to think based on those things. Well, there's one other piece of information that we know beforehand, and that led to our first.
There is a compound already in another species that it's known to be causing the stinging.
Right. So moroidin in related, and it's not just in other species, it's in related species, moroidin causes stings. He knows that. And so that leads to the original idea and what's the original idea?
The original idea would that be that we would assume that this species also has moroidin, but then they eventually find that that's incorrect, and that could be like leading into Act 2.
Exactly. So Act 2 begins with then that conflicting evidence. So we got that first step there. This is all the stuff that we thought before we found this conflicting evidence. It's the background. It's what the world looks like. World looks like before my work. And then we find something happens. We get some conflicting evidence. Now, in this case, it's a little weird because they did this work about injecting moroidin and finding that it did not cause long-lasting pain was done before Vetter's work. So I have to take existing work that's out there and shoehorn it into this form because people don't put it in this form. So it's not perfect, but you got the basic idea. So that is now part of the conflicting evidence. You could say, and that's the beginning of Act 2. So we think the world is like this, but I found that this other work had been done, and that we know moroidin, and that work showed that moroidin didn't produce lasting pain. And what's the second piece of conflicting evidence?
Moroidin wasn't present in.
And moroidin wasn't present in the
dendrocyanide.
So I hope you're seeing how interesting this is, right? By forming it this way, how interesting it is. You're hitting them at the beginning of Act 2 with a surprise. Coming back to the idea of surprise. So we thought that the world was like this, surprise, it's not. And then you're going to have a new hypothesis, and you're going to lead down to the big surprise at the end where we really understand it. So I'll use other examples from things from 3-minute theses. There's a really great 3-minute thesis on macular degeneration, and I will summarize it in this way, summarize 3 minutes in one sentence. You thought there was no cure for macular degeneration. Surprise, there might be. Surprise, even stronger, there is. And so what this guy's work was on was a new way to treat it. So, I mean, that's pretty powerful stuff, right? Take something, now that's a medically related one, so it's even more, people are more interested in it. But even here, right, even on plant research, you're talking to people who care about plants at this conference, surprise them with this new stuff that are coming in. Okay, so we got the beginning of Act 2. What is the main part of Act 2? What's the new hypothesis? What kind of stuff does she do?
It's possible that new compounds are responsible. So that would be the new hypothesis.
Yep. And then now we can kind of break that down into subsidiary hypotheses. What does she do and what she find that kind of elaborates that hypothesis? Beginning of the,
just isolated and purified these other molecular constituents from the species of interest.
Right, and you see how that refines her hypothesis now to these mini proteins. And then she has to ask, is this late eluting fraction with these mini proteins to stabilize disulfide bonds, is this really the cause? And then the rest of Act 2 is about showing that it's a cause of this. I mean, showing that, producing the evidence that it's the cause of these long-lasting stings. So then you're going through the things. So, in there you have to do a little bit of methods and stuff, but it's like, you have to say, well, we looked at voltage-gated sodium nav channels. You don't have to say how you set up the experiment. You don't have to prove that you know how to do that. Things, you just did it. I did that. I'm competent to do it. Here's the results. It's a 1 or 2 sentences. It's not a 5 minutes of your talk. And then she ends up, after all that stuff in the middle, where she does the experiments to show that these new compounds produces evidence to support the fact that they are the causative things. What is our conclusion?
So the conclusion would be that they discovered a new class of neurotoxins which could broaden the understanding of pain control.
Yeah, and they're like these really weird stuff that occurs in cone snail venoms, which are really toxic organisms. And so it's pretty, it's pretty amazing to find those things in plants. And so you can, by bringing that kind of excitement to the end of your research, you can convey to the audience through your gestures and through your vocal intonation how exciting this kind of research is, because not only are the facts and your setup, how you've gone through this exciting, but then you've conveyed it in the way that you present also. And the audience needs to say, oh my God, that's pretty cool. There's a plant with cone scale venom in it. So I know that it's one thing to do this in a workshop and it's another thing to do it with your research. Believe me, I've been there. And I would say you do not need to do it perfectly the first time. What you need to do is start. So you need to go in there and try it this time in botany, do as much of these things as you can. I would love it if you let me know how it goes. If you send me your link to when you're giving your talk or when your video is, I'll try to be there to listen to it. Especially if you've tried some of these things in it. I'd be happy to listen. Such a small group, I can say this. I would be happy to listen to your talk beforehand if you want some feedback on it. And but try, just take a step, trying to do it a little bit better each time. Find your weak points, structure your next talk so you address those weak points and do better on those. You get better really fast. But do not think you have to do this perfectly. No one is doing this perfectly. No one is giving the perfect talk. There always can be improved. It's just a way of not doing it, thinking you have to do it perfectly. So because I've said all this, that I've been there too, that I've felt like you have to do it. I have to do it better this time. I have to do it perfectly this time. And that's not true. Just has to be a little bit better than last time. So here's my analysis of this. This is one of the examples from the book. The current state of knowledge. I simplified this a lot. So we know about the plants. We know they cause long-lasting pain. And then Here's our conflicting evidence right at the beginning of Act 2. The extracts of moroidin don't cause the pain, long-lasting pain, and then when you extract it from Dendrocini excelsa, you don't find moroidin in there. So that's a big problem with the pre-existing knowledge. So it must be other pharmacological active compounds that are responsible, and then they do the research. Symptoms of the sting can be reproduced from those synthetic versions of these peptides, etc. They do all kinds of other experiments on this. And then the conclusions are the causative agents are these compounds. So that form is about 12, you should be able to do that in about 12 minutes, which is the length of this talk. It's taking a little bit more time than is here to set up the background and talk a little bit in detail about your stuff. This research is elaborate. They published it in a really good journal. It's a very long, very dense paper. You could expand this easily to an hour. And in the book, we talk about how you would do that, how you would expand these kind of short talks, I call these medium talks, medium length talks, into an hour. Short talks are about 3 minutes. You're doing about 12. I'm not going to ask you to do this now, but I want to ask you to think about how would you change your abstract. You know, if you had to rewrite it now, you might try to do that and think about how you're going to form your talk. You've focused on the title. You've got a better idea now of what your key takeaway point is, and now elaborate that point into these 3 parts: the existing world, what the conflicting evidence is at the beginning of Act 2, the things that you did to address that conflicting evidence, and then what the conclusions are, what the surprising conclusions are for the people. You are heroes, and this is the hero's journey. Every scientific talk reproduces that hero's journey. And if you were a graduate student, you might see similarities here also to your path through graduate school. The path through graduate school is a classic hero's journey. There's even a mentor there like Obi-Wan, guiding you on and teaching you those new things. And I really encourage you to think about it. If you're a graduate student listening to this, think about your work in that way. I think it'll encourage you to go on. I mean, think about all the hero stories you've heard and how they've had to struggle through to do it. And when you're having struggles, you know, think about yourself in that same light. You're going to make it through. Go back and remind you that there is a book coming out that will deal with these things. We've dealt with less than half of 2 chapters. So there's a lot more in the book about these, about presenting concisely and even presenting long, hour-long talks. There's a chapter about how you make these things into longer talks, but we talk about short elevator talks, talking about introducing yourself and networking events, as I've said, talk about posters and good poster design. So I encourage you to look at those things. I want to open it up for questions and comments here before we end.
I guess this probably depends on the project, but when we're thinking about like these 3 different acts, do you think most of the presentation should be kind of a bit around that second act, or for like a 12-minute presentation, where do you think most of the time should be allotted, I guess, or what do you suggest?
It's probably going to mostly be in the second act. It depends on how much background information your audience needs. So you have to think about who might show up for your presentation. Are you talking in a symposium? Is it a contributed talk? You know, where is it going to be in the program? Those kinds of things determine a little bit about what this audience is going to need in terms of background. If you're working in a really esoteric area that you don't think many people are going to know much about, you need to spend more time at the beginning to make sure that they're on board for that. You also want to make sure that there is a good transition between Act 1 and Act 2. So the most common problem I find in long talks, hour-long talks, is that people spend a lot of time on the background on general stuff that everyone in the audience knows already. You know, they go over the structure of the DNA, they go, you know, stuff that you don't need to tell this audience probably. And then they say blah, 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 and they're on to the, I'm literally that fast, and they're on to their research and they're deep into it. And you don't know what was the transition between, there's 4 bases and, you know, this esoteric sequencing technique that you're using now. I missed that little thing there. So make sure that transition there between, which is really the kind of transition of conflicting evidence, make sure that transition is clear. So let me say that again, about the audience and what they need to know. You don't have to repeat general stuff that this audience is going to know. You have to think about what this audience will know about your research area. Tell them the things that they do not know. You can assume, I can assume that most of my audience has never heard of the Loeaceae. I need to introduce that family to them. And I need to tell them a little bit about why it's important, why this question I'm working on is important. That's Act 1. And in my case, it's going to take a little bit longer because it's not, that stuff isn't known. And then most of my talk is going to be talking about the characters and things like that in Act 2. And then the conclusions are going to be relatively short at the very end. Act 3 is going to be relatively short in my case. It might be different for yours. It really depends on your field. As you said at the beginning, Emma, depends on your field and your audience. The transitions are really important. There's a whole nother form that we talk about in the book, popularized by Randy Olson, called the and but therefore form. The and but therefore form really focuses on the transitions between, especially between Act 1 and Act 2. That transition is very important. You know, you thought the world was this way, but there's this problem. That but really captures the audience's attention. It's very, very important. Make sure that is emphasized. That's the thing I was just talking about in the hour-long talks, that kind of but that shows there that says why I'm making this transition from the general introduction to my specific research. That needs to give a lot of emphasis there. Even though it may not be a lot of time, it needs big emphasis so it does not get missed by your audience.
First, I have a question. Are there situations or conditions where the hero's journey is not an ideal framework for a science communication presentation? Are there situations when this is not ideal?
So I'm sure that there are. I would say one of them would be a symposium talk, like we've looked at the titles for, where you may be summarizing a lifetime of research in an hour. Things. I still think that there is some application there, and we do talk about, as I say, longer forms in the book that I think might work in those kinds of cases. They're based out of what I've been talking about here. They're kind of a combination of this hero's journey and the and but therefore form. There's ways you can put those together that make, I think, really good sense for longer talks. But there's possible there in those kinds of symposium presentations. There may be other things. I mean, you'd have to find those cases. I don't think we've been doing this kind of storytelling approach in science enough to know exactly that I can say at this stage, it's going to work here, it's not going to work there. Absolutely. I think it's going to work in a lot of places.
But it's not going to work when you're defending your dissertation.
Oh yeah, absolutely. No, I mean, it's still going to be there, right? It's still going to be there. You just have to have other emphasis. It's all about the audience. One of the chapters is called "It's All About the Audience." The audience is so important. It is nice doing this workshop for the Botanical Society because we all know what the audience is going to be. We share that information about the audience. Now it's segregated in different parts of the conference, but we have a general idea what a Botanical Society audience is going to be like. The committee audience, that's a very different audience. Giving a seminar, a job seminar, that's a different audience because it's probably going to be more general. If it's a general biology department, very unlikely it's going to be all botanists, especially these days. So these are really important things to take into consideration when you're forming your talk. However, the form of the talk, there's 3 parts of it, can still be the same for these short talks. Still, what was known before? What did I change? What were the results of the changes that I made? Those are the basic 3 parts. That pretty much always works. For short talks, at the end of the video, I have to say, really, thanks for coming.